So, let's have a sound check. Can you hear me there in the back row there, guys? Can you hear me in the back row? Great. Okay. So, before we actually launch into lipid metabolism, again, I want to introduce you to something I've put together, and I would like your feedback on whether, in fact, it's a useful thing or not. If we go to ICON, let's see, is ICON in there? University ICON. Let me just, oh well, there we go. So, as you know, on ICON, there's a list of links. It's a little further down, I think, than I wanted to go. Let's go back to, what happened to the lights? There we go, let's try that. So the links is in here, and some of you, of course, have been using this already for your looking at videos. But what I want to share with you is something a little different, and that is an online textbook. So if you go into the ICON links, one of the categories now is online texts, and this says Wikipedia biochemistry text. Can you get them? Those lights seem like they're way too low for me. Where do the lights go? Okay, let's try that. Okay, so this Wikipedia text is a collection of articles that are all Wikipedia articles, but what I've done is to put them together in the general topic areas that your textbook follows. And so you can see here that these are going to touch on most of the areas that we're discussing, and you can use it as a way to cross-check on your textbook. That is, oftentimes if something in one textbook isn't quite clear, looking at another one can clarify it, but who wants to pay for 2 biochemistry textbooks? So this gives you a chance to do that. Some of these articles also go into more depth than your textbook does, and so they'll give you a handle to explore a topic in more depth if you want to. Wikipedia also allows something else, which is, in principle, you can generate a PDF of this, so you could carry it on your computer in principle. But last time I tried, yes, the Wikipedia doesn't have the computing power free. They're doing some kind of maintenance-y thing, and making PDFs takes a bit of thinking power for the computer. But you can use it in this format right away, and when Wikipedia finishes its maintenance, you can make an electronic version of it if you'd like. If you decide to use it, please give me feedback on whether you found it helpful or not. I can improve it. You can improve it. It's Wikipedia. So feel free to improve it if you can, or improve the articles in it. So any questions about that before we move on to our own topics? Okay. I don't know. Maybe some. Sorry? Question? Oh, thank you for reminding me. Eli and AJ would like to offer a help session next week. I know it's strange to think of, but we're approaching the 3rd hour exam. Next week, they'd like to offer you a choice between a Wednesday or a Thursday review session. So I'm going to ask you 3 questions. They're going to be prefer Wednesday, prefer Thursday, or either one. And if you could hold your hand up long enough for Eli and AJ to get a sense of that, that'd be great. Would you prefer a Wednesday review session? Please raise your hands and keep them up. Would be 6 again? 6 o'clock again. Hold them pretty high. Let's see. Okay, that would be a Wednesday. Would you prefer a Thursday? Would you be equally happy with both? I think that means that you guys can pick which one you want, is what I see from that. Okay? So we'll announce which one it is and send an email to that effect. Are there other questions about course management, things like that so far? Questions? I don't know why the lights are so low. Is this lower than people want them? I can raise them further. Higher? Lower? It's okay? More? Less? Oh, all right. Maybe we need to have that little clicker thing just so we can find out what we're thinking of each other. Okay. So, are there any questions about the material that we covered so far on fatty acid synthesis and fatty acid oxidation. Beta oxidation on the second carbon, fatty acid synthesis piece by piece. Any questions about those? All right, I think we were here. I'm getting ready to talk about a subject that is of practical importance and perhaps of widespread health importance. So we talked about fatty acid synthesis and fatty acid breakdown. We talked about how fatty acid synthesis actually occurs on a single enzyme in us. In bacteria, the enzymology is a little different, and it turns out that the enzyme that is responsible for fatty acid synthesis, specifically the enonyl acyl carrier protein reductase, is a target that is different enough in bacteria than in us that things can target it without hurting us. And so this compound, triclosan, is a relatively small, fairly simple molecule that is an inhibitor of this enzyme. And as a consequence, since all organisms need fatty acids, it's a very broad spectrum antibiotic or antiseptic. Some things target just the wall, and in that case, only those organisms are affected. But because all organisms need fatty acids, this is quite broad spectrum. You'll find triclosan, which is the trade name for it. Triclosan turning up in all kinds of things. If you have a deodorant soap, for instance, it probably has triclosan in it. You may see it in toothpaste. You may see it in lots of different things are likely to have triclosan in it. This is a bit of a concern because it is a good broad spectrum one, and having it in sort of promiscuous use out in the environment means that we're selecting all the time for resistance. And in fact, organisms are starting to appear that are resistant to this. So it would be desirable to be able to keep using it in a broad spectrum way in places like hospitals and clinics where there's the greatest need for it. So I would point out to you that as good and perhaps cheaper versions of things that can be used as antiseptics would include dilute bleach. Just cut bleach with a bit of water. It'll do at least as good a job as the triclosan. Sunlight disinfects things very nicely. The UV is very powerful and simple. And even canola oil. So if you want to disinfect a cutting board, to rub it down with some canola oil, will probably keep it as clean as the triclosan will. So we've focused so far on sort of a major part of the metabolic aspects of lipids, which is fatty acids, which are excellent ways of storing energy and a major component by bulk of membranes. But there's another group of molecules that are considered lipids, though their structures are rather different, that are also important, not necessarily all the time, but when they're important, they're very important. And those are ketone bodies. This is a generic name for 2 or 3 molecules that you'll see why they get called that. And the importance of ketone bodies is that when we're fasting, when we're not taking food in by mouth, and we're living off of what we've got in us already, it's a metabolic solution that allows the body to make a water-soluble supplement to glucose. Fatty acids are not water-soluble. You can't have very much fatty acid out in the bloodstream at once, or you start to deterge yourself. Ketone bodies, by contrast, don't have a detergent effect. Being able to make ketone bodies out of fatty acids spares amino acids. Now, you remember when we were doing gluconeogenesis that you had to have carbon backbones to make glucose out of. And you may remember that I said fatty acids don't provide those carbon backbones because when you burn a fatty acid, it comes in as acetyl-CoA and 2 carbons come out. We even went through the metabolism of how it is that plants can make sugar out of fatty acids, though we can't. So when you're making sugar through gluconeogenesis, fatty acids provide the energy for that, but it's amino acids that provide the carbon backbone. And so if you're dependent solely on gluconeogenesis, you have to burn up your own protein to do that. Ketone bodies allow a way for our bodies to take fatty acids and use it as an alternate fuel without having to use up protein. So it's a very important aspect of metabolism. So what's going to go It starts with 2 molecules of acetyl-CoA, so you could get those, for instance, from beta oxidation. And thiolase allows you to smoosh them together, so now you have a 4-carbon chain. This is simply acetoacetyl-CoA. Now we have a 4-carbon molecule. The energy for that was paid for out of the CoA. Sorry. Didn't do what I wanted it to do. It did what I told it to do. And then a very important molecule, HMG-CoA synthase, is going to take a 3rd molecule of acetyl-CoA and add it on, in effect, the side of the acetyl-CoA. And now you have a 6-carbon molecule, and it's 3-hydroxy, 3-methyl, glutaryl-CoA. So it's a 5-carbon molecule that kind of sort of looks like glutarate. And it's a 3-methyl, 3-hydroxy, 3-methyl, glutaryl-CoA. You can see why we often call it HMG-CoA. It's just a lot easier to say. But this is a 6-carbon intermediate, still has a handle there of the CoA molecule to manage it the rest of the way. Okay. So HMG-CoA can then be used as the source for making a molecule of acetoacetate. And when you do that, you recover one of the acetyl-CoAs. This acetoacetate can itself then undergo a couple of things. If we reduce it further, here we have 3-hydroxybutyrate dehydrogenase. It's been named for the backwards direction. So we invest in NADH, which we're probably getting by burning fatty acids. And now we have 3-hydroxybutyrate. This obviously looks like a water-soluble compound, doesn't it? It's got enough oxygen hanging off of it to make it water-soluble. Some of this acetoacetate can lose a carbon dioxide just non-enzymatically, just because it's a bit unstable. So carbon dioxide can just fall off of it and leave acetone. So here you see the 3-molecule acetone with a central carbonyl carbon. Acetone on the breath can smell kind of flowery. So someone who is making high levels of ketone bodies may have a detectable scent of acetone on their breath. Ketone body production happens in the liver. So the liver uses fatty acids through beta oxidation to provide the acetyl-CoA and to provide the energy. And then it synthesizes the ketone bodies that are then simply released into the bloodstream. And because they're water-soluble, they'll go wherever they're needed. When you get to where you're going, and one of the common places where the ketone bodies are going is to the brain. In order to use the energy in the ketone bodies, something has to happen to it. The 3-hydroxybutyrate, here you have your 4-carbon compound, your 4-carbon ketone body. You're going to oxidize it. Here we have NAD plus coming out as NADH. So now we have back our acetoacetate. So there's acetoacetate. And we want to activate that again to get it into a CoA compound. And succinyl-CoA can transfer its CoA compound onto it. That releases a succinate and an acetoacetyl-CoA now. So the succinate will continue through the rest of the citric acid cycle. We've just skipped the step that would normally provide a little bit of GTP. So now we have an acetoacetyl-CoA, and thiolase can take a free CoA, and now we have 2 molecules of acetyl-CoA ready to be fed into the citric acid cycle and produce energy the way anything else that provides acetyl-CoA can. So this is a way to ship the energy of fatty acids to parts of the body in a water-soluble form without having to use up the carbon backbones of amino acids. Any questions? Okay. This is sort of to introduce you to one of the places where some of these interesting things are happening. This is a light micrograph of electron micrograph of an adipocyte. I got confused because it's so colorful. I assumed it was a stained one, but of course people have Photoshop these days. And one of the things you see looking at this cell is it has a fairly compact nucleus and big droplets of fat. And adipocytes, or fat cells, are the place where the majority of lipids is going to be stored on an ongoing basis in a human. There is now some evidence that adipocytes, in fact, can divide and produce more adipocytes, even in adults. It's not clear that there's any fixed lifetime for a given adipocyte. If a person is undergoing an extended period of calorie deficit, the size of these fat deposits within the cell will tend to shrink, but the cell will still be there. It's still with you. In order to be able to synthesize lipids, say from sugars or from excess protein, the adipocyte has a lot of the enzymes for pentose phosphate pathway. You remember the pentose phosphate pathway produced reducing power from glucose. But in addition to the pentose phosphate pathway, it has elongases and desaturases that allow it to tweak and to fine-tune both the fatty acids it itself makes and any fatty acids that it takes in from the circulation. Adipocytes are specialized for storing fat. And an adipocyte can end up with 80 or 90% of its volume as fat. However, many cells can store fat. They just don't store it in the same proportions. So liver cells, for instance, can store fat. And in someone who has a very high fraction of their calorie content as alcohol, there may be quite a lot of fat in their liver. Muscle cells can store modest amounts of fat and can draw on it for extended, sustained aerobic exercise. You notice I say here, notice differences from book. So I'm going to give you my version of fats, diet, and heart disease. You can look at the book's version of fats, diets, and heart disease. And I'm going to put online some of the articles that I'll be talking about in connection with this. I mentioned that atherosclerosis is one of the major killers in America today. So heart disease is definitely a major medical and public health problem. The notion has been afoot that this is in some sense tied to our current lifestyle and perhaps tied to our current diet. After a lot of public health studies and extended nutritional studies, a number of findings seem to be emerging with a considerable level of confidence. Saturated fats, fats that have no double bonds, tend to raise low-density lipoproteins. They tend to raise the form of cholesterol in the blood that tends to correlate with bad stuff happening to you, like atherosclerosis. Unsaturated fats, ones that have at least one double bond, tend to lower low-density lipoproteins. This is reflected, for example, in something called the Mediterranean diet. When you look at around the rim of the Mediterranean in Spain and Greece and along the north shore of Africa, these are cultures that use a lot of olive oil. And they may be getting 40% or more of their calories from fat, but almost all that fat is in the form of olive oil, which is has unsaturated fatty acids in it. They tend to have fairly low heart disease rates. Dietary cholesterol probably doesn't make any difference. The only reason that they thought it made a difference is that in general, when you look in ordinary folks' diets, cholesterol and saturated fats tend to kind of go together. But the most striking example of this was in a very nice study done of South African egg farmers, where these rural African, black South Africans were being paid partly in eggs and eating 5 to 6 eggs a day. But apart from that, their diet was low in fat. They were getting exercise and so on. They had beautiful serum cholesterols. And it might be objected that maybe this is because of the rural South African diet. So the same investigators actually took college students who were eating a more traditionally or more conventionally Western diet and had them consuming different amounts of eggs. Again, no difference in serum cholesterol. So enjoy your eggs. Another group that sort of is a pointer to some of these interactions are the Eskimos. Eskimos eating a traditional diet have one of the highest fat content diets known for humans, but they have a pretty reasonable heart health. And it appears that the difference there is the high level of omega-3 fatty acids in their diets, that they eat large numbers of fish and marine mammals who, in order to cope with the cold water, have omega-3 fatty acids in a higher level than we do, and that this counterbalances the effect of high fat in the diet. So I'm not going to go through piece by piece and tell you where I'm disagreeing with the textbook. That's the way I see it. All right. Any questions so far? Okay. So we've talked about fatty acids. We've talked about ketone bodies. There are some other lipids as well that are fairly important, and I'd like to touch on them as well. So we mentioned, when we were talking about storage of lipids, triacylglycerols. So these are the molecules that are going to have a 3-carbon backbone and fatty acyl tails off of all 3. So we start off with glycerol 3-phosphate. You can see it's got alcohol, so this would be an alcohol without that phosphate, so it's glycerol 3-phosphate. And acyltransferase can take an acyl tail, this might be palmitate or stearate or something like that, and transfers that 2 times. You've got 2 compounds there going on, and now you have a molecule of phosphatidate. You may remember this when we were discussing membrane lipids. So this is the glycerol backbone with 2 tails and a phosphate. You might wonder where we got that glycerol phosphate from. Dihydroxyacetone phosphate can have that central carbon reduced at the expense of NADH. That's the glycerol 3-phosphate that we have up there. So here's the phosphatidate with 2 acyl tails. And we throw off the phosphate, and with another round of acyltransferase, we now have triacylglycerol. Sorry, let me go back up one more. Yeah, okay. So some of the other lipids that are of interest are the membrane lipids that make for both the lipid bilayer and that allow for differentiating between inside and outside, and for the curvature of membranes. So these are also going to be of some importance. So we're going to look at phosphatidyl ethanolamine and phosphatidylcholine. So here we're looking at sort of a generic version of either ethanolamine or choline. In ethanolamine, NX3 is simply H's. That would be an amine group there. So ethanolamine, without that at all, you'd say ethanol. With that as an amine group, it's ethanolamine. Choline is this nitrogen, but instead of having hydrogens, it's methyls. There are 3 methyls on it. So it still has a positive charge, but it's in the presence of this kind of cloud of methyl groups. So either one of them we can start off with. Invest in ATP, and now we have either phosphatidyl ethanolamine or phosphatidylcholine, depending on what we have at that end. Invest CTP, throwing away two phosphates, splitting it off to inorganic phosphate. And now we have an activated form of ethanolamine. So we have a CDP ethanolamine or CDP choline. So we've seen this trick before of having the diphosphonucleotide as the activating group. But this is now in a form that's active for transfer to something else. If we bring in a diacylglycerol, we made some on the previous slide, lose the CMP, and now we have phosphatidyl ethanolamine, phosphatidylcholine, depending on what we have at the end there. So you can see that to add the active head group to this, we're investing 1, 2, 3 high energy bonds to get there. Here's a phosphatidyl ethanolamine that we just made. If we want to make a phosphatidyl serine, it's actually quite simple. If you look at the structure of serine and ethanolamine, they look rather similar, And in fact, all we need to do is to exchange one for the other, and now we have a phosphatidyl serine molecule. It's free, unlike some other things. Here's another. Suppose you want to make a phosphatidyl inositol. You may have heard of phosphatidyl inositol. It gets used as a surfactant in foods sometimes, and it's fairly abundant in egg yolk, if I remember correctly. So here we have a phosphatidate molecule again, the glycerol backbone, the two acyl tails, and a simple phosphate up there. We're going to use CTP again to activate it. So now we have a CDP diacylglycerol. All right? It's just simply a diacylglycerol here. But this activation up in here means that this area is available for adding something in place of the CDP. The inositol group can come in and displace the CMP here. So here's the inositol. If you look at the structure of inositol, it's a polyalcohol. It's a 6-carbon polyalcohol as opposed to a sugar. And when it displaces the CMP, it leaves intact one of the two phosphates, and so now we have phosphatidyl inositol. So those are the membrane lipids. We obviously need them for membranes, and that's their principal use. There's another family of lipids that are kind of exotic. They're not nearly as abundant as either the triacylglycerides of the fat tissue or the phospholipids of membranes, but they are more powerful, if you like, or capable of making a bigger difference per gram. And those are the eicosanoids. And these are lipids that act as biological signals. Okay? Eicosanoids comes from the Greek for the number of carbons in it. So if you count them, 2, 4, 6, 8, 10, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 carbons. Icosa is the Greek for 20. And so that's where they're getting their name from, is that they all have 20 carbons, but different things have happened to them. And if we start off with arachidonate, and this is a really unsaturated fatty acid because it has 4 double bonds. All right? And you can see that the furthest double bond is at the 6 carbon, 1, 2, 3, 4, 5, 6. So this is an omega-6 fatty acid. Since we were talking about how you talk about unsaturated fatty acids, it's omega-6. And there is a very snazzy enzyme called cyclooxygenase. Cyclooxygenase uses 2 oxygen molecules and does this. And you can see if you look at this that it's done a couple of things at once. It's created a cyclic peroxide here, and it has another peroxide hanging off of there. So this is fairly severe modifications. And in the process of doing that, it's created a ring structure here at the end of the molecule. Excuse me. This peroxide is not something the body wants around for long. Peroxidase resolves it into simple alcohol. But here we have one of the important forms of eicosanoids that gets done. There are a number of other modifications that can happen if you get interested in lipid metabolism or lipid biochemistry. There's some very deep and rich stuff going on here. And in general, these eicosanoids are things that are generated locally to affect other cells in the immediate vicinity. So they're a way, for instance, for the epithelium of the blood vessel to talk to the smooth muscle, or for the epithelium of the lungs to talk to the smooth muscle of the air passages. So they're kind of local messages. They're not as big, they're not as global in their effect as insulin is, for example, or adrenaline, but they're bigger in scale than just a feedback inhibition by a metabolite. And there's a great deal of variety in these final molecules and a great deal of variety in activities. Cyclooxygenase is needed to make these. There are several varieties of cyclooxygenase, and we abbreviate cyclooxygenase as COX. So when you see COX inhibitors, they're talking about cyclooxygenase inhibitors. And some of the things that these eicosanoids are involved in are inflammation, fever, pain, some of them signal pain, and so on. And so people for millennia have had an interest in inhibiting cyclooxygenase. The oldest of them all is aspirin. Aspirin is more formally acetyl salicylic acid. Salix in here is the Latin for willow. And willow bark has salicylic acid in it, and willow bark has been known to be a pain reliever and a fever reliever for over 2 millennia. It does have its drawbacks. It does more than just inhibit cyclooxygenase. And there's been an interest in finding more specific and less damaging things than aspirin. People have said that if aspirin were introduced to the market today, it probably would not get approved because it has enough bad side effects. One of the ones you may be familiar with is acetaminophen. And one of the things you may notice about all of these things is this ring structure. All of them involve ring structures that may be acting to compete somehow on the cyclooxygenase for that ring formation. Acetaminophen is a different family from the rest of them and tends to have fewer side effects. The one thing about acetaminophen is excesses of it can damage the liver very seriously. So someone who tries to overdose on acetaminophen will not die of the acetaminophen, but they may damage their liver badly enough that their liver never survives. So it's not a good thing for that reason. Ibuprofen is more recent than acetaminophen. You've probably all had it as Advil, for instance, is one of its branded names. But it has fewer bad side effects than acetylsalicylic aspirin. These two molecules look fancier than any of these three. These are the older of them. And these were developed in the hopes that they would be more selective. That is, there are at least 2 cyclooxygenases. There may be more. And it was known that cyclooxygenase 2 was one that they would play a larger role in pain and pain. And that if we could inhibit it, you might, for instance, have a drug that would be active for arthritis patients. And you may recognize Vioxx and Celebrex. And I think Vioxx is the one that turned out to have side effects at a high enough rate to be a significant concern, so that it is now only under a license where nothing else will work for that patient. Then it becomes worthwhile to use Vioxx because the side effect is reasonable given that situation. But you can see how these all have kind of a family resemblance. And it's an example, for instance, of the idea that competitive inhibitors need to recognize the active site of the enzyme in order to be inhibitors. All right. Well, there's another important group of molecules, cholesterol and all the steroids. And one of the reasons it doesn't matter how many eggs you make is because you eat is because you make your own cholesterol anyway. Quite well, actually. And let's see how we do that. Cholesterol is, of course, quite a big molecule. It's got all those rings and stuff. And so we're going to have, obviously, some construction to do. And we'll see that some of the key steps along the way. I don't expect you to reproduce the whole thing. But we're going to start off doing some things that look very similar to what we did for ketone synthesis. All right? They're going to look very similar. They're different in where they happen. Cholesterol synthesis happens in the cytoplasm, whereas ketone synthesis happened in the mitochondrial matrix. But we're going to start off the same way. So 2 molecules of acetyl-CoA, so 2 carbons and 2 carbons. Thiolase, we throw away the energy of the CoA. Now we have acetoacetyl-CoA. We've got our 4-carbon backbone. And we invest another acetyl-CoA using HMG-CoA synthase. So this is the molecule that synthesizes HMG, hydroxymethyl-glutaryl-CoA. So we've invested 3 molecules of acetyl-CoA. So this all looks pretty similar so far, doesn't it? Different place, similar general chemistry. Now, the place where it starts to make a big difference is what we do with the HMG. So here's the HMG-CoA, and this is the place where we get something different than when we're doing ketone body synthesis. There's an enzyme called HMG-CoA reductase, and it uses NADPH in the process. And HMG-CoA reductase is going to reduce that carbon. We're going to throw away the CoA compound. And now we have hydroxy here, the methyl here, but now we have an alcohol over here instead of the ketone body, the ketone carbonyl carbon. So this is a branched 6-carbon compound, 1, 2, 3, 4, 5, 6, called mevalonate. A trivial name, sorry about that. But because this enzyme is the committed step and the rate-limiting step also for, I shouldn't say committed step for cholesterol synthesis, but it is the rate-limiting step for cholesterol synthesis. And this compound, mevalonate, we'll see used for a number of different things. But because this is the rate-limiting step, this enzyme is very highly regulated in a number of different ways. The body can regulate the actual levels of the enzyme by synthesizing it or degrading it. They can phosphorylate it to tune it on a short-term basis up and down. This is the enzyme that is targeted by all the drugs called statins. There's a whole family of molecules that are given to people with high cholesterol levels, and this is the enzyme that they're inhibiting with the statins. To the extent that there's evidence supporting this, there is some reason to think it might be helpful for people who have had a heart attack. The evidence that it's helpful to people who haven't yet had a heart attack is not as convincing. That looks pretty good, actually. So now we have a mevalonate. We've got this 5-carbon, maybe just remind you, it's a 5-carbon branched molecule. A bunch of steps happen, then magic happens. We're going to make a compound isopentenyl pyrophosphate. So let's take a look at the structure of this. Penta, we know there's 5 carbons in it. Pentenyl, double bond in it somewhere. And an iso, so it's probably branched. And that's what this is. So we have 4 of the carbons in a row with a double bond there, and the iso part is the methyl group hanging off of that. So this is isopentenyl pyrophosphate. Isopentenyl pyrophosphate is a very important precursor. We use it for cholesterol. We use it to make ubiquinone. We use it to make farnesyl, and we're going to see what farnesyl looks like and why that matters in a little bit. And although we can't make carotenoids, plants use it for carotenoids, and of course we do depend on the carotenoids. So this is a very important precursor. It gives rise to a lot of different things. This just shows you some of those things in their structural glory. Here's the farnesyl group. You can see double bond branch, double bond branch, double bond branch. So it's got 3 isopentenyl pyrophosphate groups made it up. Farnesyl is a tail, a modification put on proteins. And when we talked about the lipid-linked membrane proteins, this is one of the lipids that can get put on a protein to make it a lipid-linked membrane protein. So this is an important structural thing for attaching membrane proteins to the membrane. Here's ubiquinone again. You remember this is the electron carrier in oxidative phosphorylation in the mitochondrion. And it's got 10 copies here, 10, of the isopentenyl pyrophosphate. So if you think about it, 10 times 4 here, it's 40 carbons deep, if you like. It's almost as long as the lipid bilayer is wide. So it's quite a large side chain. Here's the beta-carotene. You can see it's closed on both ends, but it's got 1, 2, 3, 4 copies interlonged here. You can tell that it had more than that to account for the ends. All of them, all derivatives of isopentenyl pyrophosphate are branched lipids. They will always have branches at regular 4-carbon intervals. And perhaps one of the strangest of all is what happens to make cholesterol. This is a very schematic diagram of squalene. Squalene is an extended derivative of isopentenyl pyrophosphate. We can tell how many by counting the branches. 1, 2, 3, 4, 5, 6. So 6 copies of isopentenyl pyrophosphate, head to tail. It got its name squalene because this is one of the major lipids in sperm oil. And it has the interesting property of changing it between liquid and solid depending on the pressure on it, as well as temperature. And what's cute about squalene is that you can kind of fold it up like origami, and this is showing you kind of a sketch of the folding, and end up with cholesterol. So here you have the cholesterol backbone, multiple ring structures, the 4 fused rings. You can see the little methyls still hanging off, and a little bit of a tail still left that wasn't used up in the ring structure. Very elegant synthesis. Now, we mentioned that HMG-CoA was an important product of the synthesis going towards cholesterol, that it was the rate-limiting step. And so if we can control the synthesis of HMG-CoA, that's how we control the synthesis of cholesterol to some extent in the body. And you can see here a couple of the statins that are used in medical practice. And you can see here the structural resemblances in this region to HMG-CoA, especially the HMG part of it. So this is, again, the example of structural similarity for competitive inhibitors. So what is our body going to do with all of that cholesterol? Of course, it's essential in our membranes. It's one of the things that the body uses to control the fluidity of membranes. Some of the cholesterol will be turned into esters in order to store it. So if we have cholesterol that we're going to park for a while, we can add an acyl tail to it. Here's an acyl-CoA, and the energy there is sufficient to transfer it to that alcohol group. So this is now a cholesterol ester with the fatty acyl tail here in addition to the branch tail there. Cholesterol is a precursor for all the steroid hormones. There are steroid hormones obviously involved in sexual identity, like estrogen and testosterone and progesterone. There are also steroids that are involved with mineral balance, sugar balance, like the mineralocorticoids and the glucocorticoids. Those may sound familiar. Those are steroids that are used to regulate many aspects of metabolism. It's also a precursor for bile salts. So this is one of the bile salts. And you can see the kind of core here of cholesterol, but it has been oxidized at one end, so it now has a carboxylic acid there. This is a good detergent, as you can imagine. So this part is good at the lipidy bits. This part is going to be hydrophilic. And the bile salts are in the digestive fluids that are squirted into our stomachs and mixed with the food to help digest the food we've gotten and make it available to the body. So I referred a couple of times to LDL and HDL, and they act as significant modulators of the cholesterol content of cells. They were first discovered by individuals who turned out to not be able to dock the HDL part, and so we had a chance to see what in excess of they were lacking the LDL receptors. They were never able to clear them out of the blood. So we mentioned that LDLs are one of the ways that fat and cholesterol from food can be delivered to cells. So this low-density lipoprotein structure is in the bloodstream. There's a particular receptor on cells that can bind to the protein components of the LDL and then internalize it and release into the cytoplasm the cholesterol and the lipids that were in that LDL. To go in the other direction, a separate set of machinery is used. The high-density lipoprotein, HDL, it's higher density because it has more protein than lipid, and protein is denser than lipid. So the HDL is able to come in, and it docks near a component of the membrane that can transport or flip out lipids and cholesterol as the cell finds useful to get rid of it. They're loaded into the LDL, into the HDL, and then are free to go elsewhere in the body. So this is the grand overview of lipid metabolism. Gives you a chance to see all the pathways we've talked about so far in one overall view. Let's see, where should we start from? Okay. We might as well start at the top. So if you have triacylglycerols, diacylglycerol or phosphatidyate by taking off one of the tails or adding a phosphate, we can make glycerophospholipids spending some CTP. We can continue to break that down to fatty acids. Those fatty acids in bulk would be going into beta oxidation if we're going to be burning them, and producing QH2 and NADH in the process. Each 2-carbon piece does that and produces an acetyl-CoA, which can go into the citric acid cycle. We could also have used that fatty acid to make eicosanoids. Here we have acetyl-CoA. I mentioned the citric acid cycle here, producing ATP. Or the acetyl-CoA can be used in ketogenesis. Ketogenesis goes through the HMG-CoA and then to ketone bodies. Ketone bodies are especially used by the brain deeper into fasting. We're going to see more about that in a little bit. And then after arriving in the brain, they get put back into acetyl-CoA and then burnt through the citric acid cycle. Let's say we want to make fatty acids. Let's say we've had a lot of protein, a lot of sugar, more than we need at the moment. We're making more acetyl-CoA than we can burn at the moment. Then we can synthesize fatty acids. To do that, we're going to invest a little ATP to prime it to make a malonyl-CoA. And then the acyl carrier protein is going to have the 3-keto acyl. We have to reduce it a bunch because the keto is an oxidized carbon. NADPH investment to reduce it. And then that acyl carrier protein, when it's elongated to the 16 carbons, can be released as a fatty acid. That NADPH, of course, was probably coming from the pentose phosphate pathway, but that's okay. If we have enough extra acetyl-CoA to make fat, we probably have enough extra sugar to make NADPH. Acetyl-CoA is also the building block to go to HMG-CoA, not just for ketone bodies, but in the pathway going towards cholesterol, mevalonate, isoprenoids, finally to cholesterol. So HMG-CoA reductase is not the committed step for cholesterol, but it is the rate-limiting step. So, any questions about the large, fuzzy universe of lipids? I'll see you Friday then. Nitrogen.